Hey everybody, hope you're doing well. Welcome to Fix Your Gut live stream. I'm your host, John Brisson. Actually, let me bring up my website real quick. Um, you could, of course, visit www.fixyourgut.com. You can download the Optimal Health Blueprint and get supplement recommendations for both males and females. Order gut health coaching from me, or health and fitness coaching with Jason Hooper. Order Fix Your Gut. Uh, get Formline Performance's liposomal glutathione, the old bulletproof glutathione formula, the only one we recommend on the market. Get a Smidge probiotic, which is one of the safest probiotics that a person can take. There's no lactate producing strains, delactate producing strains, or histamine producing strains in Smidge. Not that some people won't react negatively to Smidge, but it's more rare than other probiotics because of that. And then Jason Hooper released a recent post on why antacids like TUM may worsen your health over time. I wrote a post about breaking down the Optimal Health Blueprint. Jason Hooper did a post on 5 reasons why you should lower your vitamin C dosage. Of course, you click up here for research for the blog, and there you go. There's over 200 blog articles that you can read for free. We're going to have a membership section soon started. And there'll be access to members-only articles, members-only streams, members-only question and answer streams, members-only podcasts with me and Jason Hooper. We've already recorded one, so that'll be coming out soon. So why don't we begin? How outdoor pollution affects indoor air quality. So we know that indoor air quality, just by most people, their houses and stuff like that, it can make them sick in general. But is there a possibility that outdoor your outdoor air quality, does it blend in with your indoor air quality? Okay. Just when you thought you could head indoors to be safe from the air pollution that plagues the Salt Lake Valley, new research shows that elevated air pollution events like horror movie villains claw their ways into indoor spaces because our houses are not airtight. The research, conducted in conjunction with the Utah Division of Facilities Construction and Management, is published in Science of the Total Environment. In a long-term study in the Salt Lake area building, researchers found the amount of air pollution that comes indoors depends on the type of outdoor pollution. Wildfires, fireworks, wintertime inversions all affect indoor air to different degrees, says Daniel Mendoza, a research assistant professor in the Department of Atmospheric Sciences and visiting assistant professor in the Department of City and Metropolitan Planning. The study is unique. Mendoza says combining a long-term indoor air quality monitoring project with paired outdoor measurements and research-grade instruments. We all know about the inversions, Mendoza says. We all know how large of a problem wildfires are, but do we really know what happens when we're inside? Mendoza, who also holds appointments as an adjunct assistant professor in the pulmonary division of the School of Medicine, is a senior scientist at the Nexus Institute, and his colleagues set up their air monitoring equipment at the United States laboratories in Taylorsville, Utah. They placed 3 sensors to measure airborne concentrations of particulate matter, one on the roof to measure outdoor air, one in the air handling room where the outdoor air comes in, and one in the office. The building uses a 100% outside air filtration system that is not typical for most commercial buildings, which usually use some amount of recirculated air. The sensors stayed in place from April 2018 to May 2019, just over a year. In Salt Lake Valley, a year's air quality events, including firework-related holidays on Independence Day and Pioneer Day, July 24th, smoke from the wildfires throughout the west that settles in the Bull Lake Valley, and wintertime inversions in which the whole valley's emissions are trapped in a pool of cold air. Through it all, all team sensors kept watch. Amid the expected events, however, a private fireworks show took place on August 17, 2018, within 5 miles of the study building, providing unexpected research opportunity. More on that later. During a wintertime inversion event in December, as air quality index outdoors reached orange and red levels, the indoor air quality reached yellow levels and stayed there until inversion cleared. In all, the pollution levels inside were about 30% of what they were outside. That's not surprising, Mendoza said. During inversions, only around 20% of the air pollution is what's called primary pollution, the particulate matter that comes directly from combustion exhaust. The rest is secondary, formed as gases undergo chemical reactions under specific meteorological conditions, and combine to form solid particulates. As soon as the air comes indoors, these meteorological conditions change. That changes the chemical environment for those particles, and they actually dissociate, Mendoza says. That's what we're suspecting is happening when these particles come into a building, and that's why we don't observe them. In late August 2018, when 3 active wildfires burning in California, indoor air pollution rose to about 78% of outdoor pollution levels. For nearly 48 hours, the research wrote, indoor air quality reached levels considered problematic for health-compromised populations and nearly reached levels considered unsafe for all populations. It's important to note, though, that thanks to the building's air handling system, the air is still safer inside than outside. The reason for the higher infiltration of particulate matter, Mendoza says, is that smoke particles are stable and don't break down in different temperature and humidity conditions. We see those particles travel straight through the system, Mendoza says, because there's no specific filtration that blocks out these particles. Smoke particles can also be smaller in size. That's why they're so dangerous for us. Utah has 2 major fireworks holidays, July 4th and July 24th, Pioneer Day. But the researchers happened to catch a signal from a private fireworks event just a few weeks before the wildfire smoke event, providing an opportunity to see how fireworks shows, both large and small, affected indoor air quality. Smoke from fireworks is somewhere between inversion, pollution, and wildfires. It contains primary smoke particles as well as the gases that can combine to produce secondary particulates, which can come from the chemicals used to produce fireworks' bright colors. On the night of July 4th, 2018, air quality sharply deteriorated once fireworks shows began and stayed in the red range. With spikes into the purple, very unhealthy range for 3 hours. Indoor air quality reached orange levels, registering about 30% of the outdoor air pollution. It was only after 8 a.m. on July 5th that indoor air quality returned to pre-fireworks levels. Researchers write, the private fireworks show on August 17th lasted only 30 minutes, and although the scope was much smaller, the smoke was still enough to raise the indoor air quality index to orange for several minutes. Even a small fireworks show did have a marked impact on indoor air quality, Mendoza says. This matters to people with respiratory challenges who can see large-scale poor air quality events like inversions and firework holidays coming, but who might find private fireworks shows an unpleasant surprise. The commercial building that the research studies is a somewhat controlled environment. Learning about indoor air quality in homes will be a greater challenge. You have kids coming in with mud or with dirt on their feet. You have vacuuming and cooking. So that's going to be our next step, as many people are spending time at home due to COVID-19 pandemic. The research will hopefully help understand what actions people can take to improve their indoor air quality. There's a lot of opportunity to reduce the pollutants that reach occupants in buildings, both commercial and residential, says Sarah Boyle, Assistant Director for the Utah Division of Facilities Construction and Management. To me, that's a great part of this work. With more research, it could point the way to protecting people indoors. So a couple of things you can do to reduce your indoor air pollution, or even air pollution that comes out, from outside sources into your house. One, you can get put plants, indoor plants. They can help improve your air quality inside your house. You gotta be careful though, because sometimes fungus and mold can grow in those plants as well, which can cause problems to the indoor air quality. Second, you can get a high quality air filter like Rabbit Air. Rabbit Air sells 2 good air filters. I have one in my house, the -A2. It's an excellent air filter. So yes, you can use an air filter to clean out the air in your house. Third, you can buy very strong HEPA filters with different particulate grades to put in your heating and air conditioner, your HVAC unit. However, the higher that the filtration for an HVAC unit for the filter, as far as its MERV rating is concerned, the more often that you will have to replace it, because if you do not replace them and they get full of dirt and hair and animal hair or whatever, they can end up straining your heating and air conditioner unit and reduce its life if you're going to use stronger MERV filters to improve the air quality of your house. So there's just a few things that you can do to improve the air quality of your house. and hopefully that will help. So let's start with the next article. Potentially harmful chemicals found in plastic toys. It has been long known that several chemicals used in plastic toys in different parts of the world can be harmful to human health. However, it's difficult for parents to figure out how to avoid plastic toys containing chemicals that may cause possible health risks to their children. We can stop buying the cheap Chinese plastic-produced toys. Regulations and label schemes are different across regions and countries, and there's no international agreement on what substances should be banned for use in toy materials. For the most part, regulations and international lists of chemicals of concern in toys focus on certain substances with known harmful properties, such as I can always mispronounce this word, phthalates, which is a plasticizer, sauce in plastic, do not cover the wide range of chemicals found in plastic toys. Research from the DTU and the University of Michigan, together with UN Environment, has looked into this important issue, analyzed data on chemical functions and amounts found in plastic toys, and quantified related children exposure potential health risks. They rank the chemicals according to their health risk and compare the results with existing priority substances lists from around the world. The study has been published with open access in the Journal of Environmental International. Out of 419 chemicals found in hard, soft, and foam plastic materials used in children's toys, we identified 126 substances that could potentially harm children's health, either via cancer or non-cancerous effects, including 31 plasticizers, 18 flame retardants, and 8 fragrances. Being harmful in our study means that these chemicals' estimated exposure doses exceeds regulatory reference doses, or cancer risk exceed regulatory risk thresholds. These substances can be prioritized for phase out in toy materials, replaced with safer and more sustainable alternatives, says Peter Feintke, professor of the DTU Management and Studies Principal Investigator. Niccolo Aressano, the study's first author and Peter's PhD student, explains that toy manufacturers usually do not provide any information on the chemical content in their toys, and toy composition databases are missing. Hence, the researchers had to collect and scrutinize information on chemical contents in toy materials based on chemical test data for specific toys in 25 different peer-reviewed studies. Niccolo further states, we've combined the reported chemical content in toy materials with material characteristics and toy use patterns, such as how long a child typically plays with the toy, whether it puts the toys in their mouth, and how many toys are found in a household per child. We use this information to estimate exposure using high throughput mass balance models and compare exposure doses with doses below which there is no acceptable risk to children. The researchers found in Western countries have on average about 18 kilograms of plastic toys, which underlies the large amount of plastic that children are surrounded by on a daily basis. Chemicals that these researchers identified of public concern for children's health, for example, widely known phthalates and brominated flame retardants, but also 2 plasticizers, butyrate TXIB and citrate ATBC, which use alternatives to some regulated phthalates. These alternatives show indications for high non-cancer risk potentials in exposed children and should be further assessed to avoid regrettable substitutions where one harmful chemical is replaced with a similarly harmful alternative. Overall, soft plastics could cause high exposure to certain chemicals. Inhalation exposure dominates overall children exposure, because children potentially inhale chemicals diffusing out of all toys in the room while usually only touching one toy at a time, Peter Feintke explains. Many lists exist and inform about chemicals of concern across product and material applications. However, one is commonly missing is any information about the levels at which these used chemicals in different applications would be safe and sustainable. Here, the researchers introduced a new metric to benchmark chemical contents in toy materials based on exposure and risk. Peter Feintke explains, since the same chemicals could be found in different concentrations across toy materials, we've estimated the maximum acceptable chemical content for all substances reported to be found in plastic toys. Such information will enable decision makers to develop benchmarks for various chemicals in different applications, but will also help toy companies to evaluate the amount of chemicals used for specific function against such benchmarks. For parents, it will continue to be difficult to avoid using plastic toys that contain harmful chemicals. So regulators include all substances and address exposure to toys that are produced outside Europe and imported into the European market. A good piece of advice for researchers is hence to reduce the consumption of plastic materials in general, avoid the use of soft plastic toys, and remember to ventilate your child's rooms well every day. In further news, periodontal disease causes elevated cardiac risk. People with periodontitis are at a higher risk of experiencing major cardiovascular events, according to new research from Forsyth Institute and Harvard University scientists and colleagues. In a longitudinal study published recently in the Journal of Periodontology, Dr. Thomas Van Dyke, senior member of staff of Forsyth, Dr. Ahmed Tawakol of Massachusetts General Hospital, and their collaborators showed the inflammation associated with active gum disease was predictive arterial inflammation, which can cause heart attacks, strokes, and other dangerous manifestations of cardiovascular disease. Again, another study proving this. For the study, researchers performed PET and CT scans on 304 individuals to view and quantify inflammation in the arteries and gums of each patient. Follow-up studies approximately 4 years later, 13 of those individuals developed major adverse cardiovascular events. Presence of periodontal inflammation was shown to be predictive of cardiovascular events, even after researchers controlled for all the risk factors such as smoking, high blood pressure, obesity, and diabetes. Importantly, researchers found that bone loss from prior periodontal disease was not associated with cardiovascular events. Patients did not have actively inflamed gums, had a lower risk of cardiovascular disease, even if those individuals had a prior history of periodontal disease, as evidenced by periodontal bone loss in their CT scans. Yep, if you take care of your teeth, even if you had issues with your teeth in the past and issues with your gums, will likely, reduce your chance of having a cardiovascular issue. It's important. It's very differently related to people who have currently active inflammatory disease, says Van Dyke, who's Vice President of Clinical and Translational Research at Forsyth. Researchers hypothesize that local periodontal inflammation activities immobilizes cells, signaling through bone marrow, which triggers the inflammation of artery, leading to adverse cardiac events. While the study sample size is relatively small, Van Dyke said the observation is significant and should be a much larger population. For people with active gum disease, seeking treatment could potentially prevent a dangerous cardiac event. If you're in the age zone for cardiovascular disease or have known cardiovascular disease, ignoring your periodontal disease can actually be dangerous and may increase your risk of heart attack, Van Dyke said. Mitochondrial function can play a significant part in serious disease. Disorders of cells' energy supply can cause a number of serious disease, but also seem to be connected to aging. More research is needed on mitochondrial function to find future treatments. A new study involving researchers at Karolinska Institute shows how an important molecule inside the mitochondria affects their function in mice and fruit flies. A study, which is published in Science Advances, adds valuable knowledge on formerly relative unexplored protein modifications. Each cell of the body is an organ called the mitochondrion, which converts nutrients in our food into energy. I mean, it's a little more complex than that. Mitochondrion is where the Krebs cycle happens, and electron transport chain, and adenosine triphosphate production. Mitochondria are an essential part of metabolism. When things go wrong, we can develop essential serious diseases. That's true. Mitochondrial dysfunction is a hallmark of a group of rare genetic disorders, but can also be observed in common diseases such as diabetes, heart disease, neurodegenerative diseases, and in the normal aging process. That's true. What research is needed is the mitochondria and how they communicate with the rest of the cell. Scientists are able to find new therapeutic approaches to improve mitochondrial function. Researchers at the Karolinska Institute, the Max Planck Institute for Biology of Aging, and Cologne and the University of California, San Diego, have now studied how the methylation of protein affects different mitochondrial processes. Methylation is a chemical modification in which a methyl group, CH3, is added to a molecule, methylation cycle, thereby potentially affecting its function. C-adenosylmethionine, SAM, also known as ADOMET, is a main methyl group donor within the cell, including inside of the mitochondria. That's true. We're interested in studying this particular molecule since the production of SAM changes in cancer when we age, says Anna Riedenberg, researcher at the Department of Medical Biochemistry and Biophysics, Karolinska Institute. By completely removing SAM from the mitochondria of fruit flies and mice, the researchers have been able to study which processes in the mitochondria are dependent on methylation. Early studies have shown that both SAM and cellular energy levels drop during aging. Our study suggests a link between these two pathways by demonstrating that low SAM levels can influence mitochondrial energy production. Yep, if you're under methylator, per the work of Dr. Ben Lynch, per the work of Dr. Amy Asco, and among others, you want to correct methylation. You want to improve the methylation cycle so that, for one thing, for mitochondrial health and mitochondrial function. This is another study that just proves that, among other things, for proper functioning of almost every cell in your entire body, and proper, if you're an undermethylator, you generally deal with elevated histamine or histamine metabolism issues. It's quite common in undermethylators. Depression is quite common in undermethylators. Dr. Ben Lynch has an excellent book covering the methylation cycle and genes called Dirty Genes. I definitely recommend anybody listening to follow the work of Dr. Ben Lynch to read his book Dirty Genes and apply it in your life. The studies identify which the mitochondrial proteins are methylated and how methylation affects them and how these modifications might affect mitochondrial function. Researchers also demonstrate the physiological consequences of the lack of such changes. However, several questions still need answering. Our study has provided an indication that some modifications can be modified by diet. We need to continue examining if we change the pathological processes for the better. This is Anna Rindenberg. So far, we've only looked at protein changes, but other molecules can be modified by intramitochondrial SAM. We have to study these modifications to get a better understanding of the role it plays. Making swimming pools safer by reducing chlorine disinfection byproducts. I'm not going to read this whole article, but you can use a copper silver ionization method to pretty much render chlorine less harmful by the byproducts that chlorine forms when used for disinfecting. And the copper and the silver ions themselves also are antimicrobial as well. So you can end up using less chlorine. You can end up having cleaner water by using the copper and silver ions. And you can reduce the formation of chlorine byproducts in swimming pools. It's quite interesting. And finally, gut microbiome implicated in healthy aging and longevity. The gut microbiome is an integral component of the body. What's important in the human aging process is unclear. ISB researchers and their collaborators have identified distinct signatures in the gut microbiome that are associated with either healthy or unhealthy aging trajectories, which in turn predicts survival in a population of older individuals, which in turn predicts... The work is set to be published in the Journal of Nature Metabolism. The research team analyzed gut microbiome phenotypic and clinical data from over 9,000 people between the ages of 18 and 101 years old across 3 independent cohorts. The team focused in particular on longitudinal data from a cohort of over 900 community-dwelling older individuals, 78 to 98 years old, allowing them to track health and survival outcomes. The data showed that gut microbiomes became increasingly unique, increasingly divergent from others as individuals age, starting in mid to late adulthood, which corresponded with a steady decline in abundance of core bacterial genre bacteroids that tend to be shared across humans. Okay, that's interesting. Because a healthy person, bacteroids make up, the gram-negative, they make up about 20% of your microbiome. Strikingly, when microbiomes become increasingly unique to each individual in healthy aging, the metabolic functions microbiomes were carrying out shared common traits. The gut uniqueness signature was highly correlated with several microbially derived metabolites in blood plasma, including one, tryptophan-derived indole, that has previously been shown to extend lifespan in mice. Blood levels of another metabolite, phenylacetylglutamine, showed the strongest association with uniqueness, and prior work has shown this metabolite is indeed highly elevated in the blood of centurions. Wow, okay. This uniqueness signature can predict patient survival in the latest decades of life, said ISB research scientist Dr. Tomasz Wilemanski, who led the study. Healthy individuals around 80 years of age show continued microbial drift towards a unique compositional state, but the drift was less absent in less healthy individuals. Interestingly, this uniqueness pattern appears to start midlife, 40 to 50 years old, associated with a clear metabolomic signature, suggesting that the microbiome changes may not be simply diagnostic of healthy aging, but they might also contribute directly to health as we age, Willimansky said. For example, indoles are known to reduce inflammation in the gut, and chronic inflammation is thought to be a major driver of the progression of age-related morbidities. Prior results in microbiome aging appear inconsistent, with some reports showing decline in core gut genre and centurion populations, while others show relatively stability of the microbiome up to the onset of age-related declines in health, said microbiome specialist Dr. Sean Gibbons, corresponding author of the paper. Our work, which is the first to incorporate a detailed analysis of health and survival, may resolve these inconsistencies. Specifically, we show two distinct aging trajectories: a decline in core microbes and accompanying rise in uniqueness in healthier individuals, consistent with prior results in community-dwelling centurions, and the maintenance of core microbes in less healthy individuals. The analysis highlights the fact that the adult gut microbiome continues to develop with advanced age in healthy individuals, but not in unhealthy ones, and the microbiome compositions associated with healthy in early to mid-adulthood may not be compatible with health in late adulthood. Wow, okay. This is exciting work, because you think, if you have a healthy gut in your young adulthood, and mid-adulthood is you have a healthy gut in your late adulthood, but that's not necessarily true. It's exciting work we think will have major clinical implications for monitoring and modifying gut microbiome health throughout a person's life, said ISB Professor Dr. Nathan Price, co-corresponding author of the paper. Huh. That's pretty interesting. A memory without a brain. Researchers identified how the slime mold Fusarium polycephalum saves memories, although it has no nervous system. Wow, okay, that's a little crazy. Having a memory of past events enables us to take smarter decisions about the future. Researchers at the Max Planck Institute for Dynamics, self-organization at the Technical University of Munich, have now identified how the slime mold Fusarium polycephalum saves memories, although it has no nervous system. The ability to store and recover information gives an organism a clear advantage when searching for food or avoiding harmful environments. Traditionally, has been attributed to organisms that have a nervous system. A new study authored by Marina Kramer and Professor Karen Elam changes this view by uncovering the surprising abilities of a highly dynamic single-cell organism to store and retrieve information about its environment. Slime mold Fusarium polycephalum has been puzzling researchers for many decades, existing at the crossroads between the kingdoms of animals, plant, and fungi. This unique organism provides insight into the early evolutionary history of eukaryotes, to which humans belong. So eukaryotes are multiple cells. In a body, it's a giant single cell made up of interconnected tubes that form intricate networks. The single amoeba-like cell may stretch several centimeters or even meters, featuring as the largest cell on the Earth in the Guinness Book of World Records. Decision-making on the most basic levels of life. Striking abilities of the slime mold to solve complex problems, such as finding the shortest path through a maze, earned it the attribute intelligent, and triggered the research community and kindled questions about decision-making in the most basic levels of life. Decision-making ability of Fusarium, especially fascinating, given its tubular network constantly undergoes fast reorganization, growing, disintegrating its tubes by completely lacking an organized center. Researchers discovered the organism weaves memories of food encounters directly into the architecture of the network-like body and uses stored information when making future decisions. Anybody else find this kind of creepy? Kind of creepy. It's very excited that a project develops from a single experimental observation, says Karen Elam, head of the Biological Physics and Morphogenesis group at the MPDS and the professor on professor on theory of biological networks at the Technical University of Munich. When the researchers followed the migration and feeding processes of the organism, observed a distinct imprint of the food source on the pattern of thicker and thinner tubes in the network long after feeding. Given P. polycephalus's highly dynamic network reorganization, the persistence of imprints sparked the idea that the network architecture itself could serve as a memory of the past. Wow. For this purpose, the researchers combined microscopic observations of the adaptation of the tubular network with theoretical modeling. An encounter with food triggers the release of a chemical that travels from the location where food was found throughout the organism and softens the tubes in the network, making the whole organism reorient its migration towards the food. The gradual softening is where the existing imprints of previous food sources come into play, where information is stored and retrieved. This first author, Mima Kramer. Past feeding events are embedded in the hierarchy of the tube diameters, especially in the arrangement of thick and thin tubes in the network. The softening chemicals now transported, the thick tubes in the network act as highway and traffic networks, enabling quick transport across the whole organism, says Mirna Kramer. Previous encounters imprinted in the network architecture thus weigh into the decision about the future direction of migration. Given the simplicity of this living network, the ability of Fusarium to form memories is intriguing. It is remarkable that the organism relies on such a simple mechanism, yet controls in such a fine-tuned manner, says Karen Alem. The results present an important piece of the puzzle, understanding the behavior of this ancient organism, at the same time points to universal principles underlying behavior. We envision potential applications in our findings as designing smart materials and building soft robots. Wow. That can navigate through complex environments. So I always got to take it to the biotechnological level, transhumanism. All right, everybody. That's it for this evening. Take care.